My name is Damir Khismatullin. I'm Associate Professor of Biomedical Engineering and the Principal Investigator of Biomedical Acoustic Laboratory at Tulane University. Today, I would like to present the data of our recent study on resensitization of breast cancer cells by focus ultrasound. If you have further questions or are interested to learn about research conducted in my lab, you can read the lab website or reach me via email or phone. Female breast cancer is the most diagnosed cancer and the second leading cause of cancer death. More than 70% of all breast cancer cases are classified as estrogen receptor positive or ER-positive breast cancers. Endocrine or hormone therapy is the first line of treatment for ER-positive breast cancer. Tamoxifen is the most prescribed hormonal drug, particularly for premenopausal women with ER-positive breast cancer. In breast cancer cell, tamoxifen prevents estrogen binding to its receptor through competitive inhibition, which shut down steroid signaling and encoding proteins that regulate cell growth. Fulvestrant is a more potent drug than tamoxifen. It degrades estrogen receptor and is prescribed to postmenopausal women resistant to tamoxifen. With tamoxifen, the treatment regimen consists of taking this oral drug daily for several years. This eventually leads to tamoxifen resistance in about 1/3 of ER-positive breast cancer patients. This chemoresistance is acquired in two major steps. First, with the help of the histone deacetylase complexes, HDACs, the chromatin becomes condensed at the ESR1 gene, which is a gene that encodes female estrogen receptor, ER-alpha. This prevents nuclear protein binding and gene expression. Second, to survive, breast cancer cells activate other parts of their genome that regulate signaling pathways for hormone-independent cell proliferation, such as MAP, P3AKT, and NF-κB. This step transforms breast cancer cells into highly aggressive cells that spread to different organs, thus causing the lethal metastatic stage of the disease. Based on this information, tamoxifen resistance can potentially be broken through chromatin remodeling, which can be induced by mechanical vibration of the cell, as demonstrated in vitro. Focus ultrasound, or FUS, is the only clinically approved technique for cancer treatment that mechanically excites tumor cells inside the body. Focus ultrasound was shown to induce chromatin remodeling in living cells by dissociating histones from DNA. The above-mentioned facts led us to the following hypothesis. FUS-induced mechanical disruption of hormone therapy-resistant breast cancer cells lead to the estrogen receptor expression induction and resensitization of these cells to hormone therapy. We anticipate that the mechanical vibration of the cell induced by FUS decondenses chromatin at the ESR1 gene and also prevents activation of hormone-dependent prosurveillance signaling pathways. To test this hypothesis, we exposed non-resistant and resistant breast cancer cells, all based on MCF7 cell line, as well as patient-derived breast cancer xenografts, to a combination of FUS and tamoxifen. We studied the effect of FUS on both the tamoxifen and fulvestrant resistant cell, as well as on the cell with mutated ESR1 gene. In most experiments, the cells were continuously exposed to tamoxifen, and they were pelleted before exposed to FUS, and after that they recultured. FUS was operated either in the continuous mode or the pulse mode. The center frequency was 1.1 megahertz and acoustic intensity was 0.7 kilowatt per centimeter square, referred to as level H4, or 0.88 kilowatt per centimeter square level H5. Both levels were below the clinical high. Several molecular assays were done with recultured cells, a live death assay to assess cell viability and apoptosis, a cell proliferation assay, RT-PCR of ESR1 gene activity, Western blots of ER-alpha, as well as histones and proteins that control alternative pro-survival, and histological analysis was done of the xenograft tissues post-high focus ultrasound exposure. In the first set of experiments, breast cancer cells were incubated with tamoxifen at different concentration, ranging from 100 to 5,000 nanomolar. FUS applied to the cell only one time. Without FUS treatment, the viability of tamoxifen-resistant FCF7 cell remained high, more than 70%, even at 5,000 nanomolar of tamoxifen. While the viability of non-resistant cells, blue line, was 30% at this high concentration. With FUS treatment, the viability of both resistant and non-resistant cells significantly reduced, and the tamoxifen dose effect was similar between these two types of cells. Thus, FUS resensitized tamoxifen-resistant breast cancer cell to tamoxifen. The similar data were obtained for fulvestrant-resistant cells. The cell proliferation data also support that resistant breast cancer cell becomes responsive to tamoxifen post-FUS treatment. The resistant cell exposed to tamoxifen alone continued to have a high proliferation rate, 96 hours post-treatment, as shown here. FUS applied to the cell pretreated with tamoxifen caused gradual reduction in proliferation rate with time for both resistant and non-resistant cells. As you can compare TAM plus H4, TAM plus H4 groups for MCF7 and MCF7TR cells. In a second set of experiments, tamoxifen was removed from the culture medium at day 1 post-FUS, or sham treatment, which means no FUS, and reintroduced into the medium at day 5, when the 70% confluence of these breast cells, cancer cells achieved. The cells in the sham groups have higher viability after day 5, as seen here, blue and red, black lines. But viability of the cells pretreated with FUS was much lower at day 5. And also, as you see, compare this green and red line, that's reduced at the same rate for both resistant and non-resistant cells. This indicates that long-term resensitization of breast cancer cell to tamoxifen was achieved by one-time FUS treatment. Our RT-PCR data point out that prior to the treatment, the ESR1 gene was much less active in tamoxifen-resistant breast cancer cell as compared to non-resistant cells or the resistant cell with the degraded estrogen receptor or mutated ESR1 gene. The FUS treatment had a slight but significant effect on MCF7 cells, regular MCF7 cells. However, for resistant cells, a very strong, significant effect at both 24 and 70 hours was achieved after FUS treatment. So with 27-fold increase in gene expression at 72 hours. Our Western blot data shows that the expression of key markers of alternative pro-survival signaling pathways such as AKT and FKB significantly reduced after tamoxifen and focus ultrasound treatment. Thus, these cells like basically the resistant cells, cannot rely on those pathways to survive, and together with restoration of ESR1 activity, they come back to hormone-dependent growth. In addition, we found that one of the histones involved in chromatin condensation, histone H3, reduced its expression in the nucleus of tamoxifen-resistant cells post-treatment with tamoxifen and focused ultrasound, as shown here. This indicates that FUS-induced changes in DNA packing and gene expression happens through loss of histones that regulate chromatin structure. To further understand how focused ultrasound breaks chemoresistance, we compare the viability of tamoxifen-resistant breast cancer cell pre-treated with tamoxifen and then exposed to continuous focused ultrasound, which is TAM plus H4 group, pulsed focused ultrasound, TAM plus PH4 group, or hit at level generated by continuous FUS, which is TAM plus WB group. It is known that pulsed FUS causes mechanical damage to tissue, known as histotripsy, while inducing minimal tissue heating. Heating the cells in a water bath had no effect on the cancer cell viability, while both continuous and pulsed FUS treatment led to a significant drop in cell viability, but with insignificant differences between these two groups. Yeah, this confirms that FUS induces resensitization of breast cancer cell to tamoxifen through mechanical disruption of these cells. In our last set of experiments, we exposed patient-derived xenografts of ER-positive breast cancer to tamoxifen and focused ultrasound. Histological analysis of treated tissue indicates that this combination treatment significantly reduced the number of viable cells as compared with sham, tamoxifen alone, or FUS alone treatment. Additionally, images obtained by scanning electron microscopy show strong mechanical disruption in the tumor tissue and cell post-FUS treatment, as shown this at the bottom pictures. Yeah. So with the formation of large membrane pores in the cell, and sometimes with the decellularization of the tumor tissue, as shown on these two pictures. Thus, by compromising the structural integrity, FUS makes breast cancer cell more vulnerable to tamoxifen therapy. In summary, this study shows that hormone therapy combined with FUS reduces the viability and growth of resistant ER-positive breast cancer cell at the same or even greater level than those of non-resistant cells. So the FUS-induced resensitization of breast cancer cell to tamoxifen occurs at acoustic intensities lower than the clinical HIFU through mechanical disruption, but not heating of tumor tissues. Third, this resensitization effect is a long-term and occurs due to multiple increase in gene expression that encodes ER-alpha. Fourth, FUS eliminates hormone-independent cell growth and cause a loss of histones that maintain chromatin structure. Our next steps in this project will be, first, to confirm that FUS causes loss of histone bound at the ESR1 gene in resistant breast cancer cell. And second, we need to do in vivo experiments with xenograft and orthotopic models of ER-positive breast cancer to confirm that FUS can resensitize hormone therapy. This study was supported by grants from National Institutes of Health, National Science Foundation, Louisiana Board of Regents, and Tulane University. Thank you very much for watching this presentation. Please post questions about this project through Symposium portal, or you can email your questions directly to me. Thank you again, and that's it for this presentation.